DNA might be called the blueprint of life, but RNA is the nitty-gritty instruction manual. RNA generally falls into two categories, coding and non-coding. Coding RNAs are messenger RNAs, which are transcribed from DNA and then translated into protein. Non-coding RNAs, however, have a variety of functions and make up over 80% of all the RNA transcripts in our body. In 2024, the Nobel Prize in Physiology and Medicine was awarded to Dr. Victor Ambros and Dr. Gary Ruvkun, the scientists who led the research that characterized a class of regulatory non-coding RNA called microRNA. In the 1990s, Dr. Ambros was studying the development of the nematode C. elegans. In particular, he was studying two mutant strains, one that was missing the Lin14 gene and one that was missing the Lin4 gene. The worms in both of these strains had developmental defects. The Lin14 gene encodes for a protein that controls the timing of developmental events, so it makes sense why worms missing this gene would have developmental defects. However, Dr. Ambros was surprised to find that the Lin4 gene did not encode for a protein at all. Instead, it gave rise to a small single-stranded RNA that was only 22 nucleotides long. Dr. Ambros confirmed his findings with Dr. Ruvkun, who was also studying the connection between Lin14 and Lin4. By comparing notes, they discovered that the small RNA derived from the Lin4 gene had a complementary sequence in the messenger RNA for Lin14. Because of this, they theorized that maybe the Lin4 small RNA acted as a post-transcriptional on-off switch that regulated gene expression of Lin14 at specific times during development. In normal wild-type worms, Lin14 would be expressed early on in development and then be turned off by the Lin4 small RNA at the correct time. Lin14 mutants would naturally exhibit the same developmental defects as a worm in which Lin14 protein expression was permanently off, since they are missing the gene entirely. But in Lin4 mutants, Lin14 expression would be permanently on, which also results in abnormal development. They published two back-to-back papers with evidence of a novel type of RNA that could regulate gene expression at the post-transcriptional level, rather than encode for a protein. A few years later, Dr. Ruvkun found another small non-coding RNA that also regulated development in C. elegans, called LET7. Just like Lin4, the gene for LET7 did not encode for a protein, but instead gave rise to a 21 nucleotide single strand of RNA that regulated the expression of many different genes that control development. What's more, LET7 is highly conserved and found in many species, from C. elegans to flies to humans. These discoveries cemented the existence of a class of non-coding regulatory RNAs called microRNA. Today, thousands of microRNAs have been reported and have been found to be involved in almost every biological process. MicroRNAs are being studied to treat genetic disorders, including cancer, autoimmune, and neurodegenerative diseases. They even have the potential to be used as biomarkers, an invaluable tool for early-stage disease diagnosis and preventative care. So a big thank you to Dr. Ambros, Dr. Ruvkun, and their research teams for uncovering this small but mighty class of RNAs, and congratulations on winning the Nobel Prize.